Hi everyone, and welcome to the Year 11 Biology Summer Work, Pathway 2, How Can We Feed the World? First of all, we want to say we're really sorry that you didn't get to sit your GCSE exams this year, but we're really pleased that you're interested in taking A-Level Biology at Carmel and that you're interested in looking at this bioenergetics pathway. We wanted to help you put all that information you've learned so far to good use and give you a little flavour of A-Level biology in a sort of real life situation. So the work we're going to go through does actually link to some careers, and if you really enjoy these lessons, then it can lead to other careers that you might be interested in researching or finding out about. This pathway in particular is looking at how we can adapt our crops to make them more efficient. How this will help increase food production while trying to decrease deforestation and potentially reduce the use of fertilisers. So we hope you enjoy the pathway and we look forward to seeing you in September. So first of all, I'll set the scene a little bit. Okay, we're going to look specifically at a very important produce called soybean. So some of you may be lactose intolerant or may be vegan, so don't drink milk products and instead drink things like soya milk. Well, soya milk comes from soya beans and we produce a very large quantity of them. But most of the countries that produce these soybean crops often have to undergo deforestation to make room for the crops. Our other issue is the graph that we've got on this page, and that's the fact that we need to double our crop production by the year 2050, just to meet the demands that we currently have. And things like veganism and lactose intolerance are actually on the increase. So the demand for things like soybeans is going up all the time. If you look at the graph, the green line at the very bottom, that's the soybean produce. The straight line is our current soybean production. The dashed line is actually the expected increase in demand. So you can see there's a difference between those two green lines. We're basically not producing enough soybeans at the moment to meet the world's demand. So what this project is going to look at is what can we do about that? How can we increase the yield of our crops? So how can we improve how many soybeans crops are actually producing so we don't have to undergo more deforestation and the current crops that we have can meet the demands that we've got. So the first thing we need to understand is this graph. So a plant gets all of its energy from light. There's a huge amount of light available around us from the sun, and the sun provides us with a huge amount of energy. But if we look in the blue circle, you can see of all that light energy that's available, plants can only absorb a very small amount. They absorb that light in a process called photosynthesis. So they absorb the light energy around us and trap that energy inside glucose. They then use the energy inside that glucose to either make itself, so to make its leaves and its flowers and all that sorts of things, and also to produce its yield, so to produce its soybeans. We then come along and eat those soybeans, or animals eat those soybeans, and they take the energy out of those soybeans. So they use the energy that's trapped inside that glucose to provide their own energy demands. We can then eat those cows, or we can eat those soybeans. But if you look really closely at the picture, you can see that the arrow going from sunlight to plants, from plants to the cow, and from cows to humans, the arrow gets smaller and smaller and smaller, so less energy is passed on each time. What that means is to meet our energy demands, we need to eat more of these foods. Could we make our plants more efficient? So actually the line that the amount of energy that they're passing on each time is more or higher, therefore we are able to get more energy out of them than we normally would. And that's what we're going to try and look at. So we're going to look at these three soybean plants in particular. So there are three leaves that we're going to look at: normal soybean leaf A, mutant soybean leaf B, and genetically modified soybean. So this is something that's becoming much more popular at the moment. They're called GMOs, genetically modified organisms. And actually, a lot of our products do contain genetically modified organisms in some way. So next time you're eating some food or a snack or a crisp or a chocolate bar or something like that, have a look at the back of the packet. It may actually say on it, no GMOs, or has traces of GMOs. GMOs means genetically modified organisms. So this plant has had a DNA from another plant or another organism inserted into it to change its genetic makeup. I will say that these leaves are not actually soybean leaves. These are just examples to show you for this example, basically. So we need to figure out which one of these plants has the highest productivity. So which one of these plants can produce the most soybean in the smallest agricultural area possible. There's a couple of things we need to know before we can do that. So this is where we're going to start. We're going to look at one of the most important biological molecules we have, and that is glucose. So we're going to look at the structure of glucose and the bonding of glucose. The first thing we need to know is, why is glucose actually important in biology? So it's a simple sugar. We call these monosaccharides. Mono meaning single, and saccharide being the scientific word for sugars. It's a chemical store of our energy. So it's a chemical energy that's stored in the bonds within the glucose molecule. By breaking down this glucose during aerobic respiration, or anaerobic respiration, we release that energy. Glucose can also be used to make other structural molecules like cellulose that we find inside plant cell walls. So it's a really important structural molecule, so it's a really important molecule that holds cells together, but its main job is to provide us with energy.
So now that we've looked at why glucose is important, next we need to look at the structure of glucose. So on the screen we have got a picture of alpha glucose, because there is two types of glucose, and the general formula is C6H12O6. So this means to say that there are 6 carbon atoms, there are 12 hydrogen atoms, and there are 6 oxygen atoms. Now you do need to make sure that you can draw this off by heart. The other skill that you need to be able to do is to label the carbons. Now this is quite easy to do once you know how to do it. So you start off with the carbon on the left hand side of the molecule, and then you just go around in a clockwise direction and allocate a specific number to a particular carbon. Now the reason why we label the carbons is so we can then specifically refer to these when we talk about bonding. On the previous slide, we discussed that the glucose that we looked at was called alpha glucose. Now, there are 2 main types of glucose that you will come across in this qualification. So there's alpha glucose and there are beta glucose. Now, they both look pretty much identical. And the reason for that is because apart from the position of the hydroxyl group on carbon 1, they are exactly the same. So if you look at where carbon 1 is on alpha glucose, you'll see that the OH, so OH is a hydroxyl group, is below, whereas on the beta glucose, the hydroxyl group, this OH group, is above. As glucose is a monosaccharide, mono means one, so monosaccharide is one sugar. Monosaccharides can be bonded together to form disaccharides, and then if you keep adding and bonding together, you will end up with a polysaccharide. Now, the process, the particular reaction for this to happen is called a condensation reaction. So you can see at the top, I've got two alpha glucose molecules that are next to each other, and their OH groups are both in line with each other. Now, two hydrogens and one oxygen will leave the molecule as H2O, so a water molecule, and that will leave the oxygen on its own in the middle. Now, oxygen has to form two bonds because it is in group 6. So then the oxygen will be bonded to carbon 1 on one alpha glucose and carbon 4 on the other alpha glucose. And this will form what's called a glycosidic bond. And then the final product at the bottom, we have got a maltose molecule. So a maltose is made out of two alpha glucoses.
So we've learned all about glucose. Now let's look at the process that makes it. Photosynthesis. So the word can actually be split up into two, photo and synthesis. Photo means light and synthesis means to make. And synthesis is a word that we use a lot in biology, so you'll see it a lot of times. So the word photosynthesis literally means using light to make. And in our case, we're using light to make glucose and water. So on the slide in front of you, you've got the word equation and the chemical equation. You should know these from GCSE anyway. But we basically use carbon dioxide and water and then the energy from light. We use that light energy to combine the carbon dioxide and the water to form glucose. The molecule that traps this light energy is called chlorophyll. So we find these green pigments inside the plant, and their job is to take the energy from light and convert it into chemical energy. We can then store that chemical energy as glucose. An added benefit is we get oxygen from this process, and that's really important for us because we need that in aerobic respiration. Underneath we have the symbol equation as well, so we need 6 carbon dioxide molecules and 6 water molecules to make glucose, which is a 6 carbon, 12 hydrogen and 6 oxygen molecule, and we have left over 6 oxygen molecules. There's something really important about the photosynthesis equation as well. So look at the 2 equations that you've got on the slide in front of you. Photosynthesis and aerobic respiration. Look really closely. Is there anything you notice? You should notice that they're actually the reverse of each other. So in photosynthesis, we use carbon dioxide, water and energy, so energy going in, to make glucose and oxygen. In aerobic respiration, we use glucose and oxygen, convert it into carbon dioxide and water, and we get the energy out. So the point of photosynthesis is to take the light energy around the plant and convert it or trap that energy within inside a glucose molecule. The point of respiration is then to reverse that process so we're actually able to get the energy out of the molecule. And that's really important. So, we know that photosynthesis is really important to trap light energy. Where does it actually happen? Well, have a look closely at the picture in front of you. You've got a picture of a leaf, and if we zoom in on the leaf, we have a picture at the top of the page. The cuticle, the epidermis, and the palisade cells. It's the palisade cells that we're really interested in. If we take one of those palisade cells and zoom in, you've got the picture of the cell on the board in front of you. These palisade cells are packed full of a little organelle, a little structure called a chloroplast. We call these structures organelles because they're little organs inside of a cell, and the chloroplast is the most common organelle inside a palisade cell. So if we zoom in on the chloroplast itself, it has quite a stacked structure. So there's lots of labels on there, but there's only one that we're really interested in, and that's the thylakoid membrane. So you can see a chloroplast has an inner and an outer membrane, and it has lots of these green lines inside it. These green lines are membranes, and the specific membrane we are interested in is called a thylakoid membrane. This thylakoid membrane has lots and lots of chlorophyll in it. So it's green because chlorophyll tends to appear green. So photosynthesis takes place in a leaf, and the organ inside, or the organelle inside that leaf that's really important, is our chloroplast. We find lots of these chloroplasts in a layer inside the leaf, called the palisade mesophyll. Inside these chloroplasts are where we find the pigment chlorophyll, and the chlorophyll is responsible for absorbing the light energy that we use in photosynthesis. This pigment actually appears green because it's able to absorb all the other different wavelengths of light, but it isn't very good at absorbing the green wavelength, so it actually reflects it. So what you're actually seeing is reflected green wavelengths, hence why to you the chlorophyll looks green. Pause the video and have a go at filling in the gaps on your worksheet, and then carry on with the video and we can check your answers. So how did you do? Did you get them all right? Fantastic. Okay, so we know that these chlorophyll are where the light energy is trapped, but we're going to look at that in slightly more detail. So you've got this picture on your worksheet as well. Stop the video and have fill it in, and then we'll go through it. So, how do plants actually trap this light energy needed for photosynthesis? Well, they use pigments. Chlorophyll isn't the only pigment, but it's one of the most important ones. So what you're seeing in front of you is a really zoomed in section of the thylakoid membrane. And they have these funnel shapes. Within these funnel shapes, or within this membrane, are loads and loads of different types of pigments. All of these pigments are responsible for absorbing the light energy, but they transfer the energy down to the centre of the membrane. And in the centre of the membrane is a little area called the reaction centre. And this is where all of the energy collects. So the job of the pigments is to absorb as much energy as possible and pass that energy down to the reaction centre. And it's the reaction centre that converts that light energy into chemical energy. And it's that chemical energy that becomes the glucose that is so important to us. So let's have a little look at how these pigments actually work. So let's have a little closer look at how these pigments actually work. These two graphs are going to be really helpful for that. The graph on the left is all about the rate of photosynthesis. So the axis up the side of the graph on the left says relative effectiveness in photosynthesis. What that really means is just rate of photosynthesis. So we can see that photosynthesis is fastest at the blue end and the red end of the spectrum. The axis along the bottom shows us the different colours of wavelengths of light and also gives us the measurement of that wavelength in nanometers. So the graph on the left shows us that photosynthesis is fastest between 400 and 450 nanometers. It also shows us that photosynthesis is fastest between 650 and 680 nanometers. So we can see that there's two peaks here. The graph on the right is an absorption graph. So this actually shows us lots of different photosynthetic pigments. We'll recognise chlorophyll, but there's actually two types of chlorophyll. So there's chlorophyll A and chlorophyll B. And then there's a couple of other pigments as well. The names of these don't really matter. You don't need to know them because we'll always give you what the pigments are. But it's just being able to use the graph. So the graph on the right up the side shows you absorption. Again, along the bottom of the graph is the wavelength of light in nanometers. What you can see is at different wavelengths of light, different pigments absorb different amounts of light. So, stop the video here, use the two graphs on the board or use the two graphs that you've got in your worksheet and answer the multiple choice questions. Then you can restart the video and we'll check your answers. So this is the answers to the multiple choice questions for you to check. So how many answers did you get right? The question at the bottom is the one that's the most important. So what is the relationship? Well, the absorption peaks match the fastest rate of photosynthesis. What this means is the more light that's absorbed by the pigments, the faster the rate for photosynthesis. Okay, so why is that important? Why do we care about how much light a pigment can absorb and how fast the rate of photosynthesis is? Have a go at this activity in your booklets and cross out the word that is wrong. So cross out the word to make the sentence correct. So, why do we care about photosynthesis? Well, the more light that is absorbed by the pigments, the faster the rate of photosynthesis will be. The faster the rate of photosynthesis, the more glucose we're able to produce. So the more of that light energy is converted into chemical energy and stored inside that glucose. The more glucose produced means we get a higher yield produced by the plant. So basically, more soybeans. The more soybeans we can get without having to clear any more land, the more efficient the plant will be. So we'll end up with a soybean plant like the picture we've got there. Lots and lots of soybeans growing. So we've looked at the 3 leaves already, but now we need to test the leaves for which pigments they actually contain. We can look at which pigments the leaves contain, and then we can make a conclusion about which leaf will have the fastest photosynthesis. The faster the photosynthesis, we know the more glucose it will make, so the higher the yield it will make, so the more soybeans it will make. So it's really important for us to understand which pigments are actually found inside the plant. To do that, we need to do a process called chromatography. So this is chromatography. You may have heard it before, you may have actually done it before. But the point of chromatography is to split substances by mass or by weight using a special piece of chromatography paper. You've got this picture on your worksheet, so add some notes to it to help you understand how it works. The first thing that we do is put a sample of each plant onto the chromatography paper. So we get our plants, we crush them down really, really small, and then we put dabs of those plants onto the paper in a specific place called the origin. We then place the chromatography paper in a solvent. What basically that solvent will then do is move up the paper. As the solvent moves up the paper, it will go through the origin line and drag the different pigments up the chromatography paper by mass. We can then measure the movement of those pigments along the chromatography paper and use that to identify what those pigments are. And you're going to have a go at this. So, the calculation you've got to learn for something called an RF value. It doesn't really matter what RF value stands for. It basically just means method to identify the pigments. We're going to measure the distance the pigment has travelled and divide this by the distance that the solvent travelled. We're going to have a go at this one together so you understand. So, we need to measure how far the solvent travelled. So we measure from the origin line up to the solvent front. In this case, that measures 120 millimetres. We then need to know how far the pigment has moved. So we measure from the origin line to where the pigment is on the paper. So we're going to divide 18 millimetres by 120 millimetres. That gives us a value of 0.12. We're then going to use the table to identify from that RF value what that pigment was. So we can see the highlighted section, a 0.15 RS value gives us that pigment. So we can now say that this leaf contains that specific pigment. Time for you to have a go now. You have this picture and you have these calculations on your worksheet, and you've got a table to fill in. So do each of the calculations, then use the table on the previous slide and work out the 3 pigments that are found in each of the plants. On the next slide, you can check your answers. So check your answers. How many of these did you get right? Did you get all of them right? Fantastic, well done. Why do we need to know this information though? Or what can we use this information for? Well, now that we know which pigments are inside these plants, we can also work out how much energy these pigment combinations are able to capture. Remember, the more light energy that these pigments can capture, the more energy can be converted into chemical energy, can be converted into glucose. The more glucose we have, the bigger the crop we can get, so the bigger the yield. So the more soybeans we can produce in the same amount of space. So we won't have to have as much deforestation. We're going to have to look at another calculation to find this out. So the next calculation we're going to look at is called gross primary productivity. This is the scientific way of basically saying how much light energy is absorbed by the chlorophyll during photosynthesis to make glucose. We already know from the beginning of this journey that there's loads of light energy available, but actually plants don't absorb that much of it. We're trying to find out which one of our leaves is the most efficient, so absorbs the most light energy, so has the most photosynthesis, so produces the most glucose. For each of the 3 leaves, we've made a picture like the one on the board for you. It tells you how much energy the plant loses, so you can figure out how much energy the plant actually absorbs. What you then need to do is turn that percentage into a value. So, for the picture on the board in front of you, there is 11,250 kilojoules of energy available. 56% of that energy is absorbed by photosynthesis. So we need to find out what 56% of 11,250 kilojoules of energy actually is. So we do 0.56 times 11,250, and that gives us 6,300 kilojoules of light energy being absorbed. That means there's 6,300 kilojoules of light energy being converted into glucose. So let's see which one of our plants is the most efficient. Pause the video, work out the absorption of the 3 other leaves on your worksheet. So check your answers. How did you do? Did you get them all right? Fantastic. Well done. So we can see that actually the first plant absorbs 64% of the light. So the first plant absorbs 7,200 kilojoules of energy. The second plant, the mutant plant, absorbs 5,400 kilojoules of energy. And the third plant, the genetically modified plant, absorbs 6,300 kilojoules of energy. Now there is a little other sneaky step in this, which is part of this calculation. This is the amount of energy that the plants absorb during photosynthesis. So this is the amount of energy that really could be made into soybeans. But the plant also uses some of that energy itself to respire and to make its other organelles. It needs to make its other leaves, it needs to make its stem, it needs to make its flowers, it needs to make its gametes. So even though that energy is potentially available, the plant will actually use up the energy to produce other things and not just the soybeans. So we need to have a look at how respiration affects this. This is another calculation. It's called the net primary productivity. Basically, the net primary productivity looks at how much energy the plant has made through photosynthesis and takes into account how much energy the plant uses itself by respiration. So the NPP is the potential yield that we have. So the NPP is the energy that's available to actually make the soybeans. The higher the NPP is, the more soybeans we will get. So pause the video here have a look at the table in your worksheet. Use the information you've collected so far to work out the NPP of the 3 leaves. This will now tell us which one of those 3 leaves is the most efficient. So this will tell us, should the farmers be planting normal soybeans, mutant soybeans, or genetically modified soybeans? Not only will it tell us which leaf they should be planting, but it also proves why they should be doing this. So check your answers. How did you do? Did you get them all right? That's great. So, we're going to use this information to basically finish the sentences on your worksheet. And you can see from the information in front of you that the normal plant has just slightly more NPP. So the normal plant produces or has the most potential energy to produce the most soybeans. So what we're saying is that around the world, people should be planting normal soybeans because they have the highest NPP, so they have the highest amount of potential energy available to produce the most soybeans, so the biggest yield. The genetically modified plant isn't far behind. So the genetically modified plant may have other advantages, which may be why people plant that crop instead. The genetically modified plant may be resistant to a pest, or may be resistant to a herbicide, or may have an advantage of taking up more nutrients. So even though the energy in photosynthesis isn't as high, it may have other nutrients that the plant needs to grow even bigger. It may be genetically modified to change the flavour of the soybean to make them sweeter or larger or rounder or saltier. It may be genetically modified to make it easier to actually collect the soybeans, so there's less damage to the plant, or it's quicker, or it's more efficient. So it depends on what the genetic modification is that might make this plant more useful. The important thing here is, though, that we have used our knowledge of photosynthesis, of glucose, and of maths to work out and prove which is the best type of soybean to be planting. So we hope you've enjoyed this pathway of how to feed the world. There's loads of links to careers here: environmental scientist, ecologist, environmentalist, journalist, researcher. The environment is a huge growth area in industries now. So many companies are investing in scientists, are investing in environmental scientists to improve their environmental impacts. Our environment and the world around us is really important, that we need to sustain it. We need to be able to support the animals and the life that we have on the planet, but we also need to live together. We also need to be able to survive and stop extinction of species. So things like this project are examples of what scientists do all the time, every day, to try and solve these problems. We hope you've enjoyed it. We hope you've learned something from it. If you have enjoyed it, why not have a go at Pathway 1 as well? And we look forward to seeing you all in September.